Hello class 12. Today we will discuss about your very important topic of your biology, and this is the development of female gametophyte, the ovule, which is also known, this process is also known as the Megasporogenesis. Means, what is Megasporogenesis? The development of female gametophyte process is known as Megasporogenesis. Right? Before this, I also taught you about the Microsporogenesis, in which I taught you about the development of male gametophyte. So Microgenesis means development of male gametophyte, where Megasporogenesis means development of female gametophyte. Okay, and in this we will discuss about the development of ovule. Okay, so let's start the topic. This diagram, you know it very well. This is the diagram of carpel, means pistil, the female reproductive part of a flower, right? So it having main three parts: stigma, the uppermost part where the pollen will land. This tubular structure is called style, where pollen tube will synthesis, and here this swollen part is known as the ovary. Inside it, the cavity is present. That hollow cavity is known as locule, and inside the locule you can see this structure, which is look like this, is known as ovule, right? So inside the ovary, ovule synthesis, and that we will discuss today, how the development of this ovule, and inside the ovule how the development of embryo sacs takes place. Okay, this we will discuss in the topic Megasporogenesis. Okay, so topic starts from here. In locule is the place inside the ovary rich of nutrition is there. Okay, and after getting that nutrition, it synthesizes the structure like it. This is the outer structure of an ovule, right? The membrane of the ovule is a outer membrane, and inside it you can see the hypodermal layer. That's I taught you in the previous topic. Inside it, the complete mass is known as parenchymatous tissue. This parenchymatous tissue is also known as the archisporial tissue, right? So all the cells which are present inside it known as either parenchymatous cell or either archisporial cells. Many cells are there which cover the whole inner mass of developing ovule. So out of all the cells, the one cell committed itself to synthesis the embryo sac inside it. That one cell is here, you can see in this diagram. This is the cell. In beginning it was archisporial or parenchymatous cell. So it get the nutrition from the nucleus, the inner mass, right? This cell will get the nutrition from nucleus and will increase in size. Now this cell is known as MMC. That means the megaspore mother cell. Yes, of course, the megaspore, that means the female gametophyte will develop from this mother cell. So that's why this is known as megaspore mother cell. Got it? Now after the development of megaspore mother cell, when it will become mature, it will take part in the cell division. Okay, and that cell division is known as meiosis. The meiosis cell division, that you know very well, it is the type of cell division in which cell divide and a diploid cell become haploid and synthesis 4 daughter new cells, right? So meiosis takes place here, and result you can see here. The 4 daughter cells are present here. The mother cell is diploid and the daughter cells are haploid. The arrangement of new cells is in a linear structure. So that's why, and they are 4 in number. So linear structure and 4 in number, call it linear tetrad. Linear tetrad. The sequence of the 4 cell is like the one cell is very near towards the callosal end and one cell is near toward micropylar end. The arrangement is like that. So the callosal end is the area which having a rich amount of nutrition, right? So the cell which is present near of it will get the maximum nutrition. Got it? And the cell which is far from the callosal end, this 4 cell, will get the less nutrition. So the cell which is present toward the callosal end will get the high amount of nutrition. एक प्रॉब्लम है, एक कंफ्यूजन होता है क्लास 12 के बच्चों को कि सर इनमें से कौन सी सेल है जो आगे चल के एम्ब्रोसेक को डेवलप करेगी. So let's finish that confusion. The cell which is near of the callosal end will get the maximum nutrition, will develop well, where the another cell will get the deficiency of the nutrition and these cells will degenerate. So here you can see in this diagram, after getting the good amount of nutrition, the cell which is present near the callosal end is still here and the rest of the cells, those are not getting good amount of nutrition and having the deficiency of it, will degenerate here. That you can see very clearly. So now it is clear, the cell which present towards the callosal end will develop the future ambrosec. Okay. So here you can see now the cell which is there is called the functional megaspore because this will become functionally active to develop the megaspore, okay, from it. So this functional megaspore is actually the future ambrosec. Okay, now this will get the good amount of nutrition, again lots of amount of nutrition, and will increase in size. That is called the elongation of cell. The cell structure increase in size, okay, and it will become mature here. Now you can see the ambrosec is here and inside it one nucleus, and you know it after the result of this meiosis cell division, now it is haploid. Okay, so when this ambrosec will become mature for the further development, it will just get entry into the another kind of cell division which is known as mitosis. The mitosis cell division is different from meiosis, where meiosis is giving the haploid cells and 4 in number, where this mitosis is given diploid cells, means the same cells. There will be no change now in the chromosome number, okay, and the result it will give 2 cells, 2 daughter cells. Okay, this is the beauty of mitosis. Okay, now in mitosis or any cell division, 2 processes are there. First, nucleus divide, and that is known as the chirokinesis, and then after the cytoplasmic divide and the new membrane synthesized for the cell. So first, what will take place in the cell division? The chirokinesis, that means the division of nucleus. So in this process, maximally nucleus will divide and no cytoplasmic division is there. So that's why this type of chirokinesis is known as the free nuclear division. Got it? Okay. So the chirokinesis will take place and the one nucleus in the mitosis cell division will divide into 2 nuclei. Okay. After that, these 2 nuclei will start to move from the center area to the polar area. Okay. These will start to migrate towards the pole. Okay, from the center. So that's why this is known as the polar migration. Means the nucleus is moving from center to the polar area. So polar migration. And after the polar migration, you can see the position of nucleus. These nucleus reached where? In polar region of the ambrosec. When these will reach into their destiny, now the another nuclear division will take place, and this is known as the chirokinesis 2. Chirokinesis 1 was here and this is chirokinesis 2, the another nucleus division. And process is what? The mitosis, means after the division we will get the 2 nuclei. So here one nuclei is dividing by mitosis process and giving 2 nuclei. The same thing will take place with this nucleus, and this nucleus will also give the same result, the 2 another nuclei. Now we having the 4 nuclei, 2 in each pole. Okay. When this karokinesis second will complete, after that the karokinesis third will start immediately after it, and each nucleus will divide again. Now after the karokinesis third, we having the 4 nuclei in each polar area. Okay, 4 nuclei are here and 4 nuclei are here. Got it now? This is the result after karokinesis third. Now out of 4 from each polar area, out of 4, any one nuclei will start to move from polar area to the central area. Okay, from polar area to the central area. You can see here in this diagram, the one nucleus is moving from polar to central area, and one nucleus is moving from this polar to the central area. And after that you can see here the 2 nuclei is present in center. Because these 2 nuclei are coming from polar region, that's why these 2 nuclei, those are present in the central area, are known as the polar nuclei, because these are migrating from where? From the polar area. Okay, so these are what? The polar nuclei. Got it now? So here you can see the total number of nucleus are 8. So 8 nucleus are present here. Remember this thing in your mind. 8 nucleus are present in the embryo cell. 3 in callosal area, 3 in micropylar area, and 2 in between of it in central area, which is known as the polar nuclei. Okay. So this is the complete division of the nucleus, and karokinesis process will complete here. So how many karokinesis you have seen here? 3 karokinesis, and after 3 karokinesis we have seen the 8 nuclei develop. Okay, after completing the karokinesis means nuclear division, now the cytoplasmic division will take place. Okay, and you can see here in this diagram, these three nuclei are covering their own cytoplasm and synthesizing the membrane inside it. That is the division of the cytoplasm, and this is known as the cytokinesis. In both area, this will take place. Here you can see the membrane is present around the nucleus, and here you can also see this membrane is present around the nucleus. That means they have synthesized their own membrane from the cytoplasm. So cytoplasmic division is going on there. That's why the process is known as the cytokinesis. Got it? Okay. Now 3 cells are present here. These 3 cells are known as the antipodal cells. Please note down this. These 3 cells are known as antipodal cells. Those are present in callosal side, callosal end area. And 3 cells are also present in the micropylar end. In between you can see in the middle area, I have drawn it with the red color. This is an egg. This cell is known as egg, and the 2 more cells are present in both sides of the egg are known as synergids. These are smaller in size as comparison that egg cell. So these are called synergids. So 2 synergids are there and 1 is egg cell. So 3 cells are here, 3 cells are here. Total how many cells? 6 cells. And you can see these 2 free nuclei present in the center, which is known as the polar nuclei. These nuclei is actually having the covering. This covering you can see, the whole covering. This is representing one more cell, which is actually the embryo cell. Okay, so this is one more cell. 3 cells are here, 3 cells are here, and 1 cell is here. So total how many cells? 7 cells. And how many nuclei? The 8 nuclei. Because of this only, the angiospermic ovule is known as the 7-cell 8 nuclei structure, and in developed ovule you will see this structure always. Okay, so here you can see now this we have a complete developed structure of ovule, right, which synthesized by the process of Megasporogenesis. This synthesized from the Megaspore mother cell, right? So this is our Megaspore. Okay, the development of female gametophyte. So with this I am ending my topic. Hope you got it. Okay, please revise your topic always. During this lockdown condition, stay at home and save your and your family life. God bless all of you.